Good morning, all. This is Professor C.P.S. Verma. Today we will discuss glycosides of your syllabus in the next chapter, and we will discuss the crude drugs under this glycoside heading of glycosides, Senna, Aloe, and almond. Almond. And the definition already we know the glycosides. It is formed with the glycon and aglycon parts. And the glycon moiety is called as sugar, and non-glycon moiety is called as aglycon. So glycosidic linkage is there, and glycosidic linkage is formed through the alpha linkage. Alpha glycosidic bond is there, and beta glycosidic bond is there, and it will in nature beta glycosidic bond is found mostly in. Then glycosides have therapeutic effects in human and animals. Then they are used in traditional and modern medicine as cardiotonic, purgative, analgesic, and anti-rheumatic, demulcent, and many other uses. These are the general therapeutic uses of the. Then classification based on the type of aglycon in glycosides. Aglycon part is more medicinally valuable, and the aglycon part decide the classification of the uh, glycosides. Like if the aglycon part contain anthracene molecule, that is anthraquinone glycoside, then steroidal moiety is there as aglycon, then it comes under cardiac glycoside, then saponin glycoside, cyanogenetic glycoside, isothiocyanate glycosides, coumarins and fumicoumarins, aldehyde glycoside, phenol glycoside, steroidal and miscellaneous glycosides. So this glycoside, and mostly the sugar part, glycon part present is glucose as a glucoside. If the glucose is the sugar part, then it is called glucoside. Then if rhamnose is the sugar moiety, glycon part, then rhamnoside. If it is digitoxose, then digitoxoside, because digitoxose is the sugar in cardiac glycoside, and this is because of the presence of digitoxose, the Killer-Killarney test, chemical test for glycosides, cardiac glycosides, is positive. Then glucose and rhamnose, then glucorhamnoside, then rhamnose and glucose, then rhamnoglucoside. These are the, if two sugars are attached to the aglycon moiety, then it is like rhamnose and glucose, then rhamnoglucoside glycoside is. Then third classification based on the type of linkage between glycon and aglycon. So already we discussed if it is carbon, then it is the linkage is through carbon, then it is called uh, C glycoside. If it is through O group, then it is O glycoside, and if it is nitrogen, then N glycoside, then O glycoside, and S glycoside in case of sulfur atom of A glycan. Then classification based on their uses, that's like cathartic glycoside, cardiac tonic glycoside, analgesic and anti-rheumatic glycoside, and anti-ulcer, etc. Then substitutes. Coronopsis strength is used by the poor people in China as substitute for costly ginseng. So the crude drug under the glycoside in this chapter is Senna. We will discuss about Senna. This what is the synonym for this? This is Senna, Acacia Senna. This is the synonym, and it is Indian Senna is also called as Tiny Valley Senna. Is the synonym of this. Acacia Senna is also given, name is given for this Indian Senna. Then it consists of dried leaves of Acacia angustifolia, known as Indian Senna or Acacia Senna. Then family Leguminosae, it should contain not less than 1% of Sennoside A and Sennoside B on dry basis. So geographical sources. This Indian Senna cultivated in Tinuveli Senna, Madurai, and other districts in Tamil Nadu, and Kadapa district in Andhra Pradesh, and Kuch in Gujarat and Rajasthan. So another heading, another point: collection, cultivation, and preparation. How you can prepare and cultivate and collect this. This is all details you can go through. Then the collection and microscopical characteristics also. Uh, we can discuss the detail. It is color is yellowish green, odor is slight, and taste mucilaginous and bitter and characteristic. And the size is in this millimeter, 7 to 8 millimeter width, and 25 to 60 length. Then shape of the leaves, lanceolate, apex cute and spine at top, and trichomes present on both surfaces. Trichomes are also there. Then this is not, it is bilateral, isobilateral leaves. These are the detailed trichomes present on both surfaces, and the details of the trichomes, it are unicellular, conical, thick-walled, warty, slightly curved at base, and then palisade tissue present on upper and lower, and then pre-cyclic fiber present towards upper epidermis and above gel. These are microscopic evaluation. Then chemical constants, Sennoside A, Sennoside B, they are renin, cassic acid, the chemical name of renin, and dianthrons as aglycon part is present. Then other anthraquinones present in Indian Senna is Sennoside C and D, renin, camphorol, aloe isorahamatin, mucilage, resin, myricil alcohol, salicylic acid, and pyrophoric acid, calcium oxalate, and tinivally glucoside. And these are the structures of steinoside A, steinoside C, and B and D. Then uses entrepreneurial glycosides absorbed first in the intestinal tract, then a glycan is separated and excreted in colon. It causes irritation and stimulation of colon. So movement, peristalsis of colon increases. Peristalsis also reduces water absorption, and soft and bulky fishes are formed due to resin and amodin content gripping occurs. Then hence drug is given with the carminative. So this is the mechanism of action, how the purgative action is taking place. Adulterants and dog Senna, and this is Cassia ovovata. This is the adulterated species. This is having the same structure and same features, but this is not having that much potency as the Indian Senna or Cassia angustifolia. Then all these details are also there. Pathocena. This is the another adulterant of Senna leaves. The spelling of leguminaceae is wrong. Leguminaceae. You have to correct it. This family, this spelling is wrong. You can correct it. Leguminaceae. Leguminaceae. Then chemical constant and address are same like Indian Senna. Then next group is aloe. The synonym of aloe is aloe, Hindi, Musabar or Kumari. Marathi, in Marathi it is called as Kurfine. Then botanical source, it is dried juices of leaves of Aloe barbadensis Miller, known as Curaco aloe, or Aloe perry Baker, known as Socotrine aloes, or Aloe ferox Miller, and hybrids of these species, Aloe africana Miller and Aloe spicata, Aloe spicatia, spicatia, and also known as Cape aloe. And the family of aloe is Liliaceae. Then cultivation collection can go through all the cultivation collection. Geographical source, it is indigenous to Southern Africa, cultivated in Iceland, Europe, and then preparation, how we can prepare the aloe, alloys, different alloys, Curaco aloe, how we can prepare, how we will treat the leaves to give, cut the leaves and then spines of the leaves and put in kerosene tins immediately after cutting, then kept tilted position in V-shape wooden troughs to drain out juice. The juice is boiled in copper pans for evaporation to obtain thick juice. Then it is poured into metal container where it is, and then Cape colors is obtained with another method similar to it, and we will get like obtained from Aloe ferox. This Cape alloys from the another species of Aloe, Cape Aloe ferox, we will get Cape alloys, and then we will prepare. Leaves are cut transversely and kept in the circular manner in basins shaped depression lined with goat skin or canvas. They are kept in this position for 5 to 6 hours till all juice exudes out and collected in goat skin. Like this is how we will make the Cape aloe juice, and we will write and we will solid. Then Socotrine alloys also we will get. It is obtained from Aloe perry. This is another species of Aloe perry in East Africa. We'll get, and the aloe we'll get through Aloe perry is named as Socotrine alloys. Then Zanzibar alloys. It is verified to Socotrine alloys. Juice is placed in skin of small carnivorous animal. When it is solidified, then packed in wooden boxes, and then further it is treated solidified. Then the description of Curaco alloys. These are the organic property. Then Cape alloys, their color, taste, and odor and fractures, different taste and different color and other thing. This for different type of alloys. Then Socotrine alloys, color, taste, and odor. This is also the details. Color is brownish yellow, opaque, masses with pasty consistency. And the taste is extremely bitter, nauseous. Odor is unpleasant. The Zanzibar alloys, color liver brown, odor characteristic but not disagreeable. Taste is bitter, texture dull, waxy, smooth. Then microscopic characteristic, when we will make the transfer section, it will show the following details. The microscopic structure of alloy powder, different alloys we will get from different species where they have the detail, another microscopic details there. In chemical constant, anthracene glycoside, it contains mainly active constant is aline. Then aline is also called as barbaline, and barbaline is C glycide because it is linked with the carbon through the carbon atom. this glucose part and aglycon part is linked through the carbon atom, so it is called C. So these are the chemical constants present in the aloe, margaline, aloe, aloe resin. These are the special tests. There are tests to distinguish between varieties of alloys. Then nitrous acid test, and this another test. This another species, different species of alloys will give different colors. Like Curaco alloys, sharpening to carmine color, and then Cape alloys, faint color, and then Socotrine and gingiber alloys, very less changes in color. And nitric acid test. Nitric acid applied to drug or its aqueous solution. Curaco alloys, deep brownish red color, and Cape alloys, brownish color, changes in green, uh, changes in brownish yellow color, and then gingiber alloys, yellowish brown color. Then Cupraline test. This is the details of the Cupraline test for different species of alloys. It will give different then uses purgative, stronger purgative than all other quinone glycoside to prevent gripping. Action carbonating can be given. It is ingredient of compound tincture of benzene and Friar's balsam. Aloe gel is used for topical application and many cosmetic. Aloe gel is obtained from inner parenchymatous cells of leaf, slightly viscous and clearly thin. It should not be contaminated with aloe juice. So these are the adulterants and substitutes. Then the last drug of your crude drug under the topic glycosides is bitter almond. The bitter almond synonym is Amygdala amara, and biological, so these are dried ripe seeds of prunus amygdalus, and family is Rosaceae. Then macroscopic, this organoleptic properties of like color, odor, taste, and size, shape. All these details of macroscopic chemical constants. It contains fixed oil, then 20% protein, bitter glycide, amygdalin, 1 to 3%, and 0.5% volatile oil. Amygdalin hydrolyzed to produce benzaldehyde and hydrocyanic acid. Hydrocyanic acid is very poisonous hence it is not used internally. Sweet almond do not contain amygdalin. So uses oil is emulsant in perfumery in preparation of bitter almond water, and then it is also used as sedative.